All right, so we're going to move on into Unit 4, which is going to deal with organisms that start with bacteria. We're going to move all the way up to plants, with plants being a relatively large focus on Unit 4. But we'll talk about bacteria and protists, and we'll mention viruses and up until we get to plants. So learning targets 1 and 2, bacteria through plants. So there's 6 kingdoms of classification in all living organisms. So you have Archaeobacteria, Eubacteria, Protista, Fungi, Plantae, and Animalia. So prokaryotes, remember what prokaryotes are. Prokaryotes are simple or more simple single-cell organisms, and they're located in both of the bacteria kingdoms, the Eubacteria and Archaeobacteria. And then your eukaryotes are your kind of multicellular or more complex celled organisms, start at Protista, and then they're also found in Plantae, Animalia, and Fungi. All right, so Archaeobacteria are bacteria-like organisms that have a different cell wall chemistry than that of Eubacteria, which means that they more than likely evolved from Eubacteria. And one way that they, besides the cell wall chemistry, that they're separated from the eubacteria is where they survive or where they live at, which is typically harsh environments. And this is why it's thought that they have evolved from eubacteria to fill these areas where most organisms cannot. They're also called extremophiles. So in the picture, you'll see a hot springs with some boiling hot water, and there are some bacteria-like organisms or archaeobacteria that live there. And most organisms cannot. Extremely cold places, extremely dry places, extremely wet places, places that is not ideal for normal life. These bacteria have developed ways to live there. All right, so eubacteria have a different cell wall, and we see a lot more diversity here in this group of bacteria. These bacteria are found in a lot more environments. When you think of bacteria, you're typically thinking about eubacteria. Okay, so this is what's going to be found on doorknobs or on desktops, or just bacteria on our skin, healthy bacteria. So this is typically when we talk about bacteria, we're talking about eubacteria, that bacteria that fills almost everywhere except for those extreme environments. All right, so we talked about photosynthesis quite a bit, and that process is very important. So remember that photosynthesis is using light to produce food, okay? Well, then there's this thing that some bacteria do called chemosynthesis, which is it's the same process as photosynthesis with the exception that chemicals are going to be used to produce food. So for example, in the deepest, darkest parts of the ocean, you have these ocean vents, and a lot of times sulfur and other gases, chemical gases, are released from the Earth's core through these vents. And there are some bacteria that live down there that use these gases to produce glucose. So it's chemosynthesis, done very similar to photosynthesis, just no light. All right, so bacterial metabolism, different types of metabolism found in bacteria. You have photoautotrophs, which are self-feeding from light, and they fix carbon using light. Photoheterotroph, they use light, but they also have to find carbon from other sources. They cannot fix their own carbon. Chemoautotrophs are self-feeding from chemicals, and they fix carbon using these chemicals. And then chemoheterotrophs are energy and carbon come from consumption of chemicals. So remember what carbon fixation is, or the fixing of carbon. That is taking inorganic carbon and making it organic carbon. Most of the carbon in the natural world is inorganic, and it has to be fixed. Bacterial reproduction, one way is binary fission. Binary fission is very similar to mitosis, which we've talked about, with the except without the cell mechanisms of a eukaryotic cell. So we're not going to have the nuclear envelope breakdown and the I'm drawing a blank. The spindle fibers attached to the centromeres and all those things. But outside of that, those specifics, this process is very similar to the splitting and replicating of genetic material and the splitting of the cell into 2 identical daughter cells. Then you have conjugation, and conjugation is not a form of sexual reproduction. And that's why we say it's not male or female combination. But it is the joining of two bacteria in order to exchange DNA. So it's similar to sexual reproduction, but it's not a form of sexual reproduction. This is just something that is pretty unique to bacteria. It's not the splitting of a cell into two daughter cells. This is the actual transfer of genetic information to make new bacteria. But again, not male and female combination, not male and female reproduction. No, this is conjugation, and it's very unique to some bacteria. All right, so bacterial shapes. You have coccus, which are spherical shapes. An example of this is staphylococcus or staph. You have bacilli, which are rod shaped, and sporilia, which are spiral shaped. Cell walls. Two different types of cell walls. We're not going to spend a whole lot of time on it. We could spend a lot of time on it, but we're going to hit both gram-positive and gram-negative real quickly. All right, so gram-negative cell walls in bacteria. These are thin cell walls, but they're usually associated with more dangerous bacteria. And the reason being is because they're pretty plain. And I know we think of thin or plain as being not hard to kill, but we have to think about how antibiotics work. And we haven't talked about that yet, but antibiotics attach and attack to certain receptors on cell walls or of these bacteria. So in gram-negative cell walls, there are not a lot of receptors or attaching points or notification center, or whatever you want to view it as, on these type of bacteria. So antibiotics typically have a hard time reacting and attaching and killing these particular bacteria. So even though there you may have some words associated with the structure of these bacteria, as you may think that they are not as strong, so to speak, they are typically more dangerous. Then your gram-positive bacteria have thick cell walls, but are a little bit more simple in their composition, which means that they are easier to kill. All right, so roles of bacteria in the ecosystem. First is decomposers. Bacteria play a humongous role in being decomposers. And again, decomposition is the breakdown of material into its most basic parts for reabsorption by other organisms and specifically plants. Bacteria play a big role in giving plants what they need, both by decomposition and, we'll talk about here in a minute, helping them fix carbon, which is right here, nitrogen fixation. Nitrogen, not carbon. Some bacteria are able to fix nitrogen, which makes it usable by other organisms, but most notably plants. Some plants actually house these bacteria in their root systems to fix nitrogen. So when we talk about fixing nitrogen, we're talking about the same thing as we did with carbon. Inorganic nitrogen has to be fixed to organic nitrogen. There are bacteria that do this, and that organic nitrogen is needed by plants as a fertilizer to allow the plants to grow and continue their cellular processes. So this is a very, very important uh, job that's done by some of these bacteria for plants, and in return, the plants get payback for living in the root system of the plant. They get access to other nutrients. They get access to water, protection. So it's a nice little ecosystem in the root system of plants. And that is the end of Learning Targets 1 and 2. So be sure you complete the lecture form that goes with this. And if you have questions, let me know. Thank you.